a time.
Yes.
Let me do it. There's on. Okay, good. Hello? Oh, yeah, it's working. Yeah. Yeah.
Okay, let's start the academic program now. And I again welcome Professor Nolte for the series of lectures. An attendance sheet is being circulated. Please sign in front of your name for lecture 1 today. We'll have a 15-minute break after lecture 1. Then you can sign for lecture 2. We will be taking a stock of the attendance every day as a mandate for GIAN. So I think let's begin this program. We again thank Professor Nolte for coming here. Also, he is sitting in room number 305 in the new chemistry building. He will be available. You can see if he's not busy, you can just knock and you can discuss with him. Also during tea break, you can discuss with him. So that's the mandate of GIAN, is we should allow many of our students to discuss, and that's what we wish you to do. And hope you have a nice time. I invite Professor Nolte for the first lecture, Introduction of Medicinal Inorganic Chemistry. Professor Nolte, please.
Well, thank you very much. And again, good afternoon, or good afternoon still to everybody. As I said, I'm happy to be here. I'm happy to see so many of you. This course will be on medicinal inorganic chemistry, as the name says there. It's going to run throughout the week. We'll have 12 lectures, which will cover different aspects of the topic. And you will see there will also be some connections. For example, in the introduction today, I will give you some keywords. And some of the keywords will, of course, come up in more depth and more specific during the week. So it makes sense, and hopefully by the end of the week, you will then also see, the connections between the different topics, and you will see things that have been achieved. But I think also if you're, aware, and if you're awake, and if you pay attention, you will also see many things that haven't been achieved yet. So there is great, of course, also great opportunity still for more research and more work. And maybe if this course is able to inspire some of you to continue in the field, then I would be, of course, super happy. As Professor Kumba already said, I'm very happy to talk to any of you, any and everybody of you. Everybody maybe will not be technically possible, but as many as possible. I will be here, of course, during the tea breaks and then also after the lectures. So you just come up to me and go ahead and talk to me, ask questions, or tell me about your own research if you want. I'd be very happy to hear about that. I also have this office that Professor Kumba indicated, and I think I will be here more or less available all time, let's say, from after lunch or so. So you are welcome to come up, and if you want to discuss or want to show me some things from your work, I'll be grateful. The way we will do this course is, of course, it's a series of lectures. That's the way it is planned, and I know that it's videotaped, so I'll try and dress up nicely and comb my hair and wash my ears when I come here. But I would also like to encourage comments and questions from you if you have. I will try and make this a little bit interactive. Sometimes I get carried away while I'm talking, and while I say things, I get carried away. If you think this is way too fast, or if you think I'm really talking bullshit, raise your hands and tell me, sorry, this is too fast. Or if you have a question, an urgent question that you think it doesn't fit at all, then raise your hands and I will interrupt, and then we can discuss this. So I'd be really grateful for questions and comments throughout the course. So don't be afraid. Yeah? Or if you think also, there are things that one should comment more on, or you would like to hear more about certain things, I can certainly always try and expand certain topics or add more to this or that. You will see while this goes along. I think the titles will be announced, so we'll have lectures every afternoon. Some are a bit longer, some are a bit shorter, but we'll try and make it fit to these sort of 2-3 hour slots in the afternoon. Okay? Right, okay. Then I think I'll start with the first lecture today. As you would expect, there is going to be an introduction, and what I will do today in the first and also the second lecture, I would just like to introduce the topic in a very general way, and I'll be looking at it from, let's say, a general medicinal chemistry point of view. So what I'm going to talk about today, to a large extent, is not specifically inorganic. It will also have aspects general of drug discovery and so on and so on. Just to give me an overview, I think we have a very diverse audience. I already heard there are some people here that even from southern India, so very, very far away. Who of you would consider yourself an inorganic chemist? Raise your hand so I can see. Who would say you're an inorganic chemist? A few. Don't be shy. Don't be shy. Well, maybe half or so. Who of you would consider yourself an organic chemist, mainly doing organic chemistry? You're welcome. I don't mind, raise your hands. Be honest. And then there is a whole lot of people in the middle who did not raise their hands yet. Would you say you're like what? Physical chemist doing analytical stuff, doing analytical work? Who's a physical or an analytical chemist? Okay. Method development, okay. And who would say, who of you, inorganic or organic or whatever, who has some experience, let's say, already with aspects of medicinal chemistry, very general? There, the hands go up in the middle, good. Okay, so do you actually have courses on medicinal chemistry? Very good, okay. So that means probably what I'm saying today, some of you will sound familiar. I hope I will be able to add at least aspects of inorganic chemistry if you are sort of a traditional, let's say, traditional organic medicinal chemist, maybe there will be some aspects which might still be new to you or unknown, or you look at it at least from a different angle than you normally would. So I hope there will be something in it for you. Okay, so if you look at this, then of course this is a periodic table of the elements, at least the shape of the periodic table of the elements. But if you go into a drugstore or into a pharmacy or even a major drug company, many of the molecules that you can buy and that you would get and that your normal drugs are made of will be made of only a handful of elements. And of course, 95% or so of it, or even more, are organic compounds. So they will contain elements like carbon and nitrogen and oxygen and hydrogen, of course. There are some which have phosphorus. Fluorine is a very recent element. So I heard about 1/3 of all new drugs on the market contain at least one atom of fluorine. So this maybe should be bigger now, maybe sort of twice the size or something. But lo and behold, you see that many of the drugs that are on the market are really either on the right-hand part of the table, and of course they have hydrogen. And what of course is striking, that there is a large part of the periodic table that is completely white. And elements of the periodic table which are not used at all or used very, very rarely, let's say, in drug research. And of course, if you're an inorganic or an analytical or so chemist, you don't just want to look at those handful of elements, but you want to look at the whole periodic table. And I guess when you look at things from a very inorganic perspective, I think what I would like to do during this week, we will not touch all of the elements here, but what I would like to do is at least give you a flavor of some of the properties of some of the elements in this dark blue part of the periodic table. So we will have many examples throughout the week that draw on compounds from this part of the periodic table, and you will see how we can use some of the special properties that these elements have to have, advanced drugs or possibilities for things like targeting or some specific activity. So let me start, before we go into details and I give you specific compounds or so, let's just go into some of the principles of medicinal chemistry in general, and this is going to be the first part that I will talk about this lecture here. It's just going to be general on medicinal inorganic chemistry, drug discovery in general, and then we'll see how inorganic chemistry ties into this. So this I took from a well-known textbook of medicinal chemistry, and this is a definition. It says, this textbook said the medicinal inorganic chemistry is about the discovery, the design, and the development of new drugs. That's wonderful. So we want to make new drugs and we want to save mankind. Fine. There are a few things here: discovery, design, and developments. And I think it makes sense to just spend a few seconds and think about those and think about what those keywords actually mean. For example, and I put some thoughts here, that people use and that are also taken from typical textbook sources. So discovery, of course, means the identification of new compounds. And very many of the drugs that you are using, or advanced drugs that are in clinical use, are either directly from natural sources. For example, microorganisms produce penicillin, a well-known antibiotic, or they are modifications of compounds that were derived from natural sources. So a lot of inspiration in this field comes from microorganisms, from plants, from marine sources. Or alternatively, we can also discover compounds by just looking at synthetic compounds that you have made, or your colleague has made, or we have made, and you can try and then see whether there are more, interesting compounds that you can obtain from screening those compound libraries which were created by chemical synthesis. I will come to these compound libraries in a second. You will come back to that. But it can either be natural or unnatural. Either one is good as long as you discover something interesting. Some compound, say, for example, with an interesting activity. And then, next thing, once we have what we call a lead compound, we need to design and we need to work on it. And this would typically be by chemical synthesis, and I guess that's why all of you are here and while we are chemists, in order to make things, to improve things, to change the properties, to adapt to certain needs. And of course, nowadays, this is what we call structure-activity relationship. So we try and investigate which particular features of which compound, do this particular function. Or it can also be done with the help of a computer. There is quantitative structure-activity relationship. You try and put numbers on it. And very often, of course, people now also use molecular modeling or docking studies or ab initio calculations or DFT calculations to get a better understanding of their compounds. So very much of this design aspect is related to chemical synthesis and or computer-aided design, so doing calculations. By the way, I think I didn't ask, is anybody here who considers themselves a computational chemist? Not a single person, but you all work with computers, right? Oh, you are. You sort of half. Okay. But you all work with computers, of course, and if you think now your mobile phone, basically can do amazing things in relation to what maybe 20 years ago would have to be done by a huge computer in a room. Now your mobile phone can do much more than that. So also this computer-aided design and computer help in computer and drug design, of course, have taken off through the roof. And a lot of the work that we do and also a lot of the things that you see in my talk will at least partly be based on computational studies. Okay, so we talked about discovery and design, and clearly there is the question of development. And development, I would like to go into a little more detail. We have a few more things here that we really need to discuss. So one thing which we typically do first in modern drug discovery is we have to identify a target. We have to decide, select, what do we want to work on, And the target can be, of course, a disease. So you can say, I want to work on malaria, or I want to work on cancer, or I want to work on, I don't know, name anything you want. So that would be something that first you have to decide on. And then, of course, in that field, if you say, okay, I want to work on malaria, you need to decide on what kind of target do I want to work. I mean, there is the mosquito that transmits malaria, and then there's the various stages of the disease, and then, your liver may be affected, or your red blood cells may be affected, or you can, maybe you have another idea where you can work. So identifying a target is already quite difficult. Typically, could be a protein or a receptor, or in the case of malaria, you will see it's maybe some precipitate of heme derivatives that you identify as your target to do something about the disease. Of course, you have to have an idea, if I pick this protein or if I pick this target structure, let's say in general, is my drug going to be able to work on it? Yeah? And that is a very good decision. I mean, that's not always easy to say. For example, even if you know that, okay, I'm working in the case of malaria on these heme crystals that, make this malaria pigment, you have to have some kind of idea, how can I work on this? How can I touch this? How can I get my hands on it? Okay, and then this is actually more or less a scientific project here, or something that you would also do in academia. You can say it's an academic exercise to a large part. This third point here is something that a drug company would do. And unfortunately, in some cases, unfortunately, the drug company will do this very, very early when they decide what to work on. So for a drug company, there has to be an economic benefit of what they're doing. We will see that you are going to invest a lot of money in drug development. We're talking about millions and even billions of US dollar or euros, or I don't know what it is, like tenths of millions of rupees, probably, something like this, hundreds of, never mind. Oh, a lot of money, yeah. And of course, at some stage, the company wants to see revenues, want to see a benefit from what they're doing. And this is a major problem. For example, if you imagine we are talking about a very rare disease, maybe a disease which, let's say, 10 people in India have. No company is ever going to work on this. You are going to treat, in the best case, 10 people if your project is successful. You are investing millions of dollars for treating a handful of people. And this is a major problem. I mean, the people are affected and the people are ill, and of course they deserve treatment, very clearly. Yeah, every patient deserves to be treated. But for major drug company, there is not going to be an economic benefit. They're not going to work on it. So there are now other initiatives. There are government-funded initiatives. There are foundations for specific diseases. So it's not out of the world. Yeah, you can still select this disease, you can still work on it, you can still try and heal the people, but it will never be done in a big company. Other topic is tropical diseases, and I guess you are very much aware of this. And if you go to, for example, Southern Africa, or if you go to Southern America, you have some tropical diseases where thousands and even millions of people can be affected. But again, no big drug company is going to work on it, not because there aren't many people. There are many people who are affected. But the people who are affected have no money to pay for the medication. And I guess India has partly solved this problem by government funding, and some projects which will help to research on it and even drug development on tropical diseases in India. But again, for a company, it's not likely. Oops. What is this? So for a company, it's not likely to be a successful project. Okay. We'll come to some of that later on. For example, antibiotic development is going to be a problem because even though many people have antibiotic infections or bacterial infection, I should say, you only take the drug maybe for, 5 days or 7 days or maximum 10 days, and then you're done. So there is no long-term revenue. Whereas if you work on something like neurodegenerative diseases, for example, Alzheimer, many people affected, and the people likely have to take the medication for a very, very long time. So if you're a company, you would rather give a drug to a patient who needs that drug for years than 10 days. So this, you have to keep this in mind also when you look at, for example, recent admissions of new drugs to the market. They are dominated by diseases like cancer. Most big drug companies have large program on neurodegenerative diseases, Parkinson, Alzheimer, and so on. There are huge programs on things like obesity in the US. Now, this is, of course, if you look at sort of average American, you can clearly see it's a problem, and you can earn a lot of money because America is a rich country, but it's not a disease that I would say is as bad as, I don't know, let's say cancer or so. I mean, people should just eat more healthy, and that would kill the problem, right? But it's easier to earn money for a drug company. So keep in mind this aspect here, and unfortunately, it's something you have to think about, and I guess if you're a responsible scientist, maybe in your later career, you will actually be able to do something about it. But it definitely requires government policy, and it definitely requires other things than just pure academic research. All right, okay. Now, let's say we have identified our target. We have convinced the CEO of our company that, yes, this is a lot of money in this project, so it's worth working on it. And then there are a few aspects that we have to do when we do drug development. We have to identify a lead structure. This is the sort of the starting point for our compound. And then there are a few keywords here that some of you will know if you have already done medicinal chemistry, so I might actually go through it just a little bit faster. There are two keywords that you have to be familiar with. One is pharmacokinetics, and this is defined as the effect of the body on the drug. So how the body metabolizes and takes up the drug. And the keyword or the abbreviation that is used is ADME, ADME, and ADME stands for absorption. How does the drug get into the body? Distribution, let's say once you take it as a pill, how is it distributed to all your body, to your arms, to your fingers, to your legs, to wherever your disease is? And then it's typically metabolized, very often in the liver. First pass, if you take something intravenously, the blood goes right through the liver. A lot of the drugs are already metabolized at that stage. So that's metabolism. And then finally, of course, the drug is eliminated. So these are the 4 steps in drug, and that's called ADME. And that's all pharmacokinetics, as we say. The opposite of it is pharmacodynamics. That's the other way around. This is actually the pharmacological effect that we want the drug to have. So this is the good thing here. This is what we want. Pharmacodynamics is what we want. But of course, again, it has to do with biodistribution. So for example, if you have a drug and that is very lipophilic and that is stored in lipids, all the fat in your body, then it might be a different thing than if it's actually in the water, in the aqueous phase, and in your body. So biodistribution is very important. It has to do with this, of course, biodistribution. You talk about the structure, the polarity of your compound. There may be metabolites involved. Yeah? Very often, for example, the drug itself isn't active, but some later metabolite is active. You will see that in many, many cases throughout the week. And of course, then there are things like interaction with the target site. The stereochemistry may be very important. If you have a chiral compound, one isomer may be active, the other one may not be active, things like this. So all these things are summarized under the term pharmacodynamics. Bioavailability is something that has become more of a focus recently, and basically what it means if you take a drug into general circulation. So let's say it's in your blood. You get an injection and it's in your blood, just to simplify things. And then your target, for example, is a solid tumor. I hope not. I mean, I hope you're not affected, but let's assume, for a second, you have a solid tumor in your stomach. And you imagine all the drug is in your blood. And the question is, how much of the drug actually gets into the tumor, which is solid and in this particular place in your body? Yeah? So you can imagine, this is an important question. If you imagine you have 7 liters of blood in your body and the tumor maybe is this size, that would already be very big. Yeah? 7 liters versus this much, biodistribution, bioavailability is very low. Unless you have a clever idea that makes the drug come from the blood into the tumor and then you can cure the tumor. So this is clearly a very important question, and we will see in some metal compounds we actually might have advantages here where we can use this to increase the bioavailability. All right, okay, so these are some key aspects that you would need to know. Another word that is in green here that will come up several times is going to be a prodrug. This is that the active agent, the active metabolite, for example, may not be what you get. So you may get an injection of a drug, it goes into your liver, it's metabolized, and then what comes out of your liver is actually what is the effective agent in the end. You'll see many examples throughout the talk, either by accident, you have a metal complex, it's transformed into something else, and then this something else is active, or you can use the concept as a concept for delivery, for stabilizing your compound, for increasing bioavailability, for example. You can use this prodrug concept. You can say, I'm designing a prodrug to increase the bioavailability, and then this is metabolized in the tumor, and you will see some examples. We can use pH, we can use enzymes, we can use redox chemistry to do this on purpose. So we're making prodrugs on purpose, and you will see, I would say really, almost every day of the week you will see examples for this prodrug concept. Let's go through the traditional phases of drug development, and I guess if you have a medicinal chemistry background, you have seen some of it. I'm just going through this, and this is based, many of the slides, this one here as well, many of these figures are actually taken from a textbook by Jim Dabrovic. Oh, by the way, I should say, there'll be reference section at the end of each day, or of each lecture rather, there's going to be a reference section. So, if you want to read up on it, there'll be some hints where you could have a look. So this is taken from an American textbook, which means it's based on the American model of drug approval. Now I should say, I'm sorry, I'm really unfamiliar with the process in India, but I would assume that many of the steps of clinical development for drugs in India are relatively similar. Yeah? There may be some specialities. There are certainly some specialities here for the European Drug Agency, the EDA, but this is the FDA model, and we can just go through it because it exemplifies what you have to do. We, as researchers, are in this phase here, typically the preclinical phase. We have a new compound, we start the testing, we do some biological tests, let's say DNA interaction, maybe we work on cell lines already, maybe we have a model where we use bacterial infections already. Yeah? So we do all these cell line, mode of action studies. If we are really advanced, then we might have some tumor xenografts in mice. So you take, for example, in cancer research, a tumor from a human, you implant it into mice, and then you study the work on this. You're already in a living system, as opposed to cells here. This is already an animal. So you can already study metabolism and pharmacology because you're injecting something into the mice. And of course, it's not human metabolism, it's mice metabolism. There is a difference. But you're already somewhere here. Yeah? But this is still all preclinical. And now let's say you have something where you think this is a good compound, it seems to work, it seems to be active. Okay, I want to go on. So you have to file an application for an investigational new drug. And in this moment, you have to go to the government agency and disclose what kind of compound you have, what you think their mode of action is. How about the toxicology? You have to identify different species, say, okay, I already worked in mice. I gave it to some hamster. I gave it to some, I don't know, let's say rats, or maybe even a pig or a chimpanzee or something. So toxicology you have done, and you have to outline how your clinical trials will be conducted. Which hospital am I going to go to? How many patients are going to be treated, and so on. And because you are talking about human, let's say, experiments in some way, you're going to give your compound to humans, you have to involve the institutional review board. So there's going to be an ethics committee, which is comprised of doctors, of clinicians, but also of patients and maybe a lawyer or a judge or so on the panel. And they will look at your drug and they will judge whether what you are proposing, what you're saying, is actually ripe enough to be given to patients. So patient safety comes in already at this stage here. Yeah? And of course, you know this very well. There have been incidents when patient safety was neglected, and then already during clinical trials here, patients were dying. And of course, that's not what you want. You can never predict, of course, but you can do your best. So patient safety comes in at a very early stage. And in fact, these so-called, there are 3 phases of clinical trials, phase 1, phase 2, phase 3, and then it goes on to approval here in the end, and it gets to the market here. But those 3 phases have distinct purposes. Phase 1 is all about safety. So you have studied your compound before in animals, mice, rats, maybe a pig, maybe a hamster or something. Yeah, but they were all animals. And of course, human metabolism is different. So the compound is taken and given to healthy volunteers, very small numbers, 5, 10, 20 maximum, something like this. Maybe up to 100 is said here. Maybe it's more like in the order of 20. Just to see, are there any adverse effects that we didn't expect? So the first person gets it and is very closely monitored. If that person is healthy, nothing happens, we say, you're fine. Metabolism is examined, excretion, some side effects are observed, and so on and so on. Nothing happens, your compound is safe. You can go on to phase 2 clinical trials. This now is really all about effectiveness. So now you are talking about patients. And again, these are all volunteers. So the patient is asked, here is a new drug on the market. Would you be willing to be included in a study? And the patient can say yes or no. You'll see there is a problem here in just a second. Yeah? But this is patient volunteers who get treated, and then the effectiveness of your new drug is established. And of course, we're also looking at safety questions and short-term side effects, something that you didn't see maybe in the small numbers, because the incident may only be 1 in 100 or 1 in 500 or 1 in 1,000 or so. Yeah? So we are beginning to see this only if we raise the numbers. Now, the point I want to make here is something that is some ethical issues that you have to consider and think about. Let's say if you have something that isn't so harmful, let's say something like the flu, or maybe like a bacterial infection that isn't very harmful. You have a new antibacterial drug. Then typically what happens is if you are a patient volunteer, you can say, okay, I'm going to take it. And then, of course, you have to compare. So you're giving it typically to another group of patients who are getting something that looks exactly like what you're given, but that has no effect. Solution of water or of sugar or salt or something. It's called a placebo. Yeah, placebo has no effect, but looks pretty much the same as your drug. This is fine if you have a bacterial infection. You can see, let's say the left half is giving the drug and the right half doesn't get the drug, and then you are healthy within 3 days and you are healthy within 6 days, by your own immune system, you're healing. So you can say, okay, the drug was effective. The time until the patient had no symptoms anymore, 3 days versus 6 days, clearly the drug is effective. Very simple case. Think about something like cancer. If you have a tumor, of course, any patient who has a tumor has a right to be treated. So you can't do this experiment. You can't say, I'm giving you the drug and you're not getting anything. That would be totally unethical. You would all be dying from your tumor. Yeah? So that can't happen. So what you have to do is you have to say, all of you have the, well, I'm just taking you to make it, visible, right? Hopefully not for you. So all the tumor patients have a right to get the best possible treatment. Yeah? Anything else, anything less than that would be unethical. So here's the problem in terms of especially cancer research. If you have a new drug, and you think this could possibly be useful, you can only give this to a patient when you're reasonably sure that you will have a pretty good effect, and you want to compare it always to the best treatment on the market. And this is a very high bar, because you're not just getting anything that works, but you want to have anything that is better than the best current treatment, which is like the top achievement. And you want to aim your first shot, you want to be already top achiever. This can be quite tricky. So what happens in this case is very, very typical in cancer research. The first group of patients that get your drug are patients that are already very, very, very ill. And anything else has been tried, and they can't be healed. And then they will get a chance to get the new treatment, because you're thinking, well, maybe there is a slight chance we can still save them. But you see what the problem is, because your drug might actually be quite effective. It might be quite good, but on, let's say, patients in an early stage of the disease, you might actually have a better drug, but you can't treat these patients initially, because there is a good treatment already, and you don't know, am I going to be better or worse? And if there is a chance that you're worse, it's a difficult question. So it's just something to keep in mind. We will talk a lot about cancer research this week, and tomorrow, the day after tomorrow, even the day after that, partly will be cancer research. And just keep in mind when we talk about clinical developments that typically in terms of clinical studies, we are aiming relatively high. There may be other diseases where it's much easier to get a drug moving forward. For example, in diagnosis, you can always say, okay, I'm giving this new drug, see what kind of pictures I get, and then I'm giving the established drug to the same patient to compare my results. So the patient isn't getting anything worse. He or she is always getting the standard treatment, and you get the standard pictures, and you can compare, is my new imaging agent, for example, is it going to be better or worse? In cancer treatment, in particular, you don't have that possibility. Okay. Fair enough. So this is phase 2, effectiveness, patient volunteers. And then you have phase 3, which is the final one. Let's say you pass through all this already. You are now in phase 3. You're very close already to market approval. Now it's about optimizing things. You already established my drug is safe. It's relatively effective. Now you want to optimize your application. These so-called double-blind studies. You compare different established treatments. Maybe you're doing a combination therapy, my compound plus something else. Is that better than the gold standard in the field? And so on and so on. You might think about different populations. People from South India may have a different metabolism from people from Himalayan region, for example. This is already pretty good. Not many compounds make it. If you think of numbers, I would say maybe you still have, let's say, 10 compounds here. You have maybe 100 compounds in phase 1. You have thousands of compounds in preclinical phase. And here, at the end, you have only one compound to the market. So even during phase 3, quite a number of compounds still fail. So maybe 50%, it may actually be more. And you already had a long, long, long investment here. Very much before that. And still, there's a fair chance that your compound will fail here, because by the end of the day, the double-blind studies, you won't see a better effect than the established treatment on the market. You might also have, for example, severe side effects in a small patient number, and this will cause your compound to fail. If you're discovering something that kills the patient, then your compound is very likely out of clinical trials very quickly. Okay, But let's say you were successful. You have a compound that is on the market. You think everything perfectly finished, looks really, really promising. You file what is called a new drug application. So the company, the sponsor, the company, the scientist, whoever, this is typically big companies now, must provide all the data that they have. And if they withhold some data, like there were, for example, severe incidents, people dying or being severely affected, but you're lying about it, then, of course, you're legally liable. Yeah? So you really have to be honest about all the tests you did. And this is a question, and I guess you have the same discussion in India, we certainly have it in Europe, about making all data available. The drug companies, of course, they try to work on the statistics here. We were talking about populations, we were talking about, double-blind studies, who do you include, who do you exclude, all these numbers. And I mean, you're a very smart scientist. You all can work with Excel. You can all do statistical tests. There is a certain way here of manipulating your data to make things look slightly better or slightly worse, depending what you want. And you all know this. I mean, you all have done scientific experiments. About being open and being frank and providing all the data at this stage here is crucial. Yeah? You can imagine the company has already invested hundreds of millions of dollars to get to this phase. Yeah? So they're really desperate to make the step to the market. On the other hand, the patients, they want to have all the information and they want to be able to assess, is this really going to be better, or is it just about the company trying to save their money? Right? So here is the approval phase. It goes to an agency in India as well as in Europe as well as in the US. And then finally your drug is admitted to the market, and then patients can get a treatment. Now, a critical issue here is patent protection and patent duration. If you have a new compound, of course, you have it patent protected. Your patent begins to run as soon as you file the patent. And typically in the US and in Europe, the patent duration is 20 years. And it can be extended slightly, but let's say it's in the order of 20 years or so. Now, this whole process here, preclinical up to phase 3, may easily be, I mean, this is a couple of years, yeah? And then this is certainly can be 6 to 10 years or so. So now we're talking about something that can easily be 12 to 15 years or so until you actually reach market. Yeah? So the remaining, 5, 6, 8 years or so, you have to gain back, you have to earn all the money that you invested here. Because after that, it's out of patent, and you can still sell your drug, but likely the price is going to be much lower because there'll be generic compounds on the market or generic drugs on the market using your compound. So this is actually an issue that has become more prevalent because we are now demanding more and more things in all these clinical phases. For example, talking about patient safety, talking about metabolism, talking about things like this. We demand more and more data from the company, which makes it more and more costly, and the time takes longer and longer and longer. And at the same time, the duration where the company can earn back the money is actually relatively small. The question, who pays for this whole process or parts of it, is really critical. We were already talking about rare diseases. Now, this is in a case where we have enough patients, but still the drug may be quite expensive. I recently read a piece about new tumor therapy where companies use immune cells from the patients, which are genetically engineered and then given back to the tumor patients. We are talking about per treatment, per one dose, if you like, taking cells, modifying, giving them back, about $500,000 for one treatment. $500,000 US. Imagine that amount of money. How much research you could do with that amount of money? How much PhD you could fund? Probably the whole room. Yeah. So there is a lot of money, and of course it's a question, is society, is your government, are you personally, who is willing to pay for this, and who should be paying for it if we demand all these things here? I'm not going to make comment on this too much. I'm just giving you some things to think about. Yeah? These aspects are not particular for inorganic drugs. I think, in fact, we have actually some advantages here for inorganic drugs. Yeah? But it's just something to think about when you're, for example, when you're sitting in a conference and you're wondering, saying, why is everybody talking about Alzheimer and about cancer and not about, tropical diseases and malaria and so on? There are reasons for this in this process. Yeah? Okay, fair enough. Let's look at this. We're slowly coming to some more inorganic aspects. Let's look at this maybe from a slightly different perspective. I've taken this here from a review We were talking about these preclinical and phase 1 and phase 2 and phase 3 trials, but of course there is a longer time span here. It starts by target identification. We talked about this. Compound screening we talked about. This may easily be more than 10 years, and we talked about patent issues. Now, the thing I want to point out, very often when we as inorganic chemists come into this process, typically we have a compound in hand or a class of compounds or a group of compounds in hand. We're entering this phase here, the compound screening phase. Very often in inorganic chemistry, we don't really work identification first, but we come in with a compound, and while we try and go ahead, we try and actually find this. So we're moving our compound ahead and looking back and trying to identify what does the compound work on. DNA is an obvious target. We were previously, one of your colleagues, I was talking about protein targets, for example. Yeah. So we are trying to establish this in a very early phase here. So we are looking at this in some detail, and we'll talk about inorganic aspects in particular as we go along. I'll talk about some more things that are maybe related in some extent to inorganic chemistry. We want to have compounds with a very high chemical diversity, and then we do some kind of screening, some kind of assay, if you like, in order to identify our lead structures. So we start here with a compound screening, and then we have a preclinical testing of a lead compound, and then we move ahead. So this library here should be as big as possible that we have a fair chance of identifying at least one or two or maybe a class of lead compounds. So this is typical, and this is true for inorganic compounds, but it's also true for organic compounds. We talk about things like chemical diversity. So how different are the compounds from one each other? Is it, for example, just a benzene derivative, which has a chlorine, a bromine, a fluorine atom attached to it? Or is it going from a benzene to a very complicated, three-dimensional, heterocyclic compound? You can clearly see diversity here is bigger. And of course, as in many cases, as in humans, as in classes, as in government, diversity is actually good. If you have larger diversity, you have a better chance of having the right lead compound or having a hit. The lead compound which we identify is selected compound with a proven and desired activity, and then we will hope and develop this further. Very often, the lead structure has the right activity, but it doesn't yet have the right properties. For example, terms about solubility, bioavailability. We talked about pharmacokinetics, so how the compound is metabolized. This is not taken into account when you have a lead structure. The lead structure is only the most active, most promising compound to start with. And very often, your final drug will be quite different from the lead structure. But this is where we begin working. Okay, and then the assay can be anything that is biological testing. We'll have quite a number of different things here. Talking about diversity, talking about compound libraries, I already hinted at this. Many of the things that are on the market are mostly organic compounds. Yeah? So they're really not very diverse at all in a way. They are what is synthetically available. Even in the big libraries that pharmaceutical compounds have, there are huge white spots because some structures are just chemically very difficult to assess. And other structures are very easy to assess. For example, if you think about simple aromatic substitution on a benzene ring or mono-substituted benzene, let's say you have an aniline or something like this. Yeah? There are certain positions when you do an electrophilic, you know this from organic chemistry, when you do electrophilic substitution, there are preferred substitution patterns. Yeah? It's either ortho and para or it's meta, depending on your substituents. And this will limit your library. If you start from an aniline, then you will only get a certain class of aniline derivatives. That's it. And the other ones, the ones that aren't preferred, won't be in your library because they're not easily accessible synthetically. Same is true, of course, for inorganic compounds. Yeah? There are reactions which work well and classes of compounds which work very well. And then there are others which are much more difficult to assess. Yeah? For example, we'll talk about metal tricarbonyl, rhenium tricarbonyl fragments. They will always be facially oriented, right? So they will all be pointing, the free carbonyls will all be pointing in the same direction because that's very easy to make. The one where they're in a meridional fashion doesn't exist. So there's no compound or barely exists. So there's very few compounds that have this kind of substitution pattern. Synthetically not accessible. Okay. as I said, there's many things to think about here. Some holes were actually identified and filled. You can use computer analysis to identify holes in your library. You can use smart algorithms that tell you this compound and this compound are similar and these aren't similar. And if you take many similars and they're just in one spot and then there is, no similar ones up here, you can try and synthesize them. So computers come in here very handy, but we're still synthetically limited to some extent. What can we do to maybe alleviate this limitation? Well, this is where inorganic chemistry comes in. If you think of chemical diversity, and you compare a simple carbon center, and this is a tetrahedral 4-coordinate carbon, and you compare this to a metal complex, which is octahedral, has 6 substituents. You all know very well what is going to happen. I mean, this is the most diverse we will ever get. Carbon typically does not have more than 4 substituents. End of story. Yeah? What does it mean? You can have 2 isomers if you have 4 different substituents. You can have 2 isomers. You know this. It's the R and S isomers. And basically, this makes up all of organic chemistry. And all of the natural products that are out there are made up by the 2 isomers of carbon. Now take an octahedral metal complex. You can have 6 different substituents, not just 4, 6. It's 2 more. And that doesn't sound so much. But if you take 6 different substituents, then you can actually have, who knows how many different isomers you can have? Octahedral, 6 different substituents. Who thinks it's 5? Who thinks it's 500? Who thinks it's 50? Are you not thinking? Who thinks it's less than 10? Hands up. I'm going to ask less than 10 and more than 10. Who thinks it's less than 10? Who thinks it's more than 10? Yeah, it must be the rest. I mean, there must be 85 hands in the air. So again, who thinks it's less than 10? Come on, raise your hands if you think it's less than 10. 6 different isomers. Okay? And who thinks it's more than 10? Who thinks it's more than 20? One person. No, a few more, okay? Who thinks it's more than 30? Still one person. Who thinks it's more than 31? You know the answer, don't you? I think it's actually 30. I haven't drawn them all, so maybe if you work out more than 30, we'll find. But anyway, we're talking about, this is from a textbook. So if the textbook is right, it's exactly 30. 6 times 6, you're thinking, right? But I don't think that's quite true because, it's 6 times 5 times 4 and so on and so on. I think that's more likely. Yeah. So if you permutate all of them. Okay. Anyway, it's 30 different isomers versus 2 different isomers. Can you imagine what an increase in diversity this is? This is 15 times more. 15 times more. Okay. So it's as if you had black and white people, and then you have 15 different varieties, and 15 times more. So you have 30 different Indians, from south and middle and, all the different states. You know what I mean, okay? So this is like India, and this is like Europe or the US, okay? All right, okay. So you don't mind political jokes, right? I really like India. I love India, so, nothing against Indians, yeah? Okay. But anyway, you see there is a huge variety here. So there's really something we can gain if we look at inorganic aspects here. Okay. We do have quite a high chemical diversity when we think about new chemical compounds. We'll talk about different stereochemistry, 6 different isomers. And they are octahedrals, so basically the rules that R and S, they don't quite apply. So, you have to have different rules here, and you can talk about very different stereochemistry, for example. But if you're talking about inorganic compounds in general, we are not just talking about molecular compounds. I mean, this is still, in a way, a molecular compound. This may be, a molecule, this is a molecule. But if you talk about inorganic compounds or inorganic materials in general, you can talk about totally new entities. For example, things like carbon or other nanotubes, things where you can have something you can put in. And you can seal it again, you can open it again. You can attach things to the surface of a carbon nanotube. You can, for example, is anybody working on metal organic frameworks, MOFs? Do you know what MOFs are? Porous materials where you can absorb things into or you can attach things to the outer side. So suddenly you are talking about what we call now materials chemistry, nanomaterials, for example. You talk about drug delivery. You can use such entities. So beyond simple small molecules that have a function, in organic chemistry now we can also have all sorts of inorganic materials. We can have layered materials. We can have porous materials. We can have all kinds of nanomaterials which we can use in medicinal chemistry. And you can see what a big diversity arises from the fact that we have all these wonderful inorganic materials. Main thing, of course, is you want to look, are they toxic? Do they have adverse side effects? Do they actually do what I want, or is it just imagination? So many, many questions still to be asked, but I guess you can see that if we think about these things, there are massive opportunities for research in inorganic chemistry here. One thing that we want to look at in great depth during this week here will be reactivity. I will go into this in much more detail in some later lectures when we talk specifically about the reactivity of inorganic compounds. For example, redox activity, for example, photophysical properties of inorganic compounds. So we really can harness here properties from inorganic compounds that are quite unique. And we'll be talking about this aspect here in particular in detail. There is a famous statement, this is probably 50 years old or so, that says, we talk about inorganic compounds, the organic ligand makes the drug. So that is to say, there are cases where we have a metal ion and then some ligands around it, and this particular person, I guess he or she was an organic chemist, says, oh, it really doesn't matter which metal is in the middle, it's really the outside the organic framework that makes the drug. And of course, this statement assumes that the metal has only a structural and it is unreactive under physiological consequences. And we will see examples where this is true, where really the ligands make the properties of the drug, but there are many, many examples, and some are given here, where really this is not true at all. Yeah? Where it's really the metal and the metal alone, or the metal, ligands around it, but really the metal that makes the activity. For example, some of you may have heard that lithium salts are very potent drugs for treating depression, manic disorders. Yeah? And basically what people are getting is literally something like lithium carbonate. Yeah, it's in some fancy tablet, so it doesn't look like lithium carbonate, but basically what's in there is a lithium salt. Yeah, it could be lithium chloride if you want. It's just that lithium carbonate is better soluble and easier taken up, that's all. So here you have a drug where clearly the lithium is the active ingredient, and obviously there is no organic ligand at all. And we'll talk a lot about tomorrow in my first lectures. We'll talk about this one single compound, Cisplatin, a very famous, very successful anti-cancer drug. Again, look at the sum formula. Ammonia, chloride, platinum. Not a single atom of carbon in it. So there are obviously compounds that are very, very, very inorganic. Brackets, no carbon at all. Brackets closed. Yeah? Very, very inorganic. And they are potent drugs, which means that there must be something special about the metal. The metal must have some property whatsoever that makes these compounds, and there are just a few examples here, that makes these compounds very, very good drugs. Okay? All right. I'm not going through this in great detail. This slide, I think, will come up again. But this is a classification of metal-based drugs that is based on an article that appeared in Nolte Transaction by my friend and colleague Enzo Alessio from Italy. And Enzo has tried to categorize the different metal-based drugs on the market. Yeah? And he has put certain classes here. And actually, this is what Enzo wrote in his article, and this is my own addition to his classification. So you can already see this is already evolving, yeah? And this classification is going on. So we have cases where the metal has just let's start here, where the metal has just a structural role. 6 different ligands, certain spatial arrangement, that's it. And we'll see examples here in my talk. I'll talk, for example, about this class of compounds. We have cases where the metal has a functional role. For example, cisplatin, the compound I just showed you, is such a case here. And we'll talk about all these things here in great detail. This will be a large part of the lecture. We also have cases where the metal alone is just a car. It's just like a vehicle, like an auto rickshaw for a certain active compound, an organic compound that is the active compound. I'll also give you an example here in this case, for example, hypoxia-activated cobalt complexes. So we'll talk about this auto rickshaw cases here as well, where really the important person is in the rickshaw, yeah, and the metal complex is just the vehicle. And then this one here, metal complexes as catalysts. Those of you who do organic chemistry or inorganic or organometallic, you all know this, that of course many metal complexes are very, very potent catalysts in drug design, but also making organic molecules. Okay. I have just a few more slides to go through, and then I think we are supposed to have a break. Is that right? And then we go on to the second part. Maybe just give me just a few more minutes now, and we'll be a bit shorter after the break. So these are some classical examples, and we'll go, though all of these will come up later, so you don't really need to remember all of this. Yeah? It's just the compounds that will come up. I have put together for a textbook some kind of timeline for medicinal inorganic chemistry. And also this timeline here, this slide you will see a couple of times again when we actually treat single items, or we start by treating single items in this timeline. I just want to highlight the fact that we are down here. This is NZ 1993, but of course now we are down here 2017, so you could add many more here, but let's see, we are down here. But we start about 4,000 years ago. So even, way before Christ, way before our time started, there were already magical uses, for example, of gold, like gold amulets that, you would put on your chest if you had a chest pain or things like this. So, I mean, this is not really science, I can say, right? So I think you will agree this is more like mystical and, sort of things. But already, this is like already 3,000 years ago, people realized that for certain diseases, it is good to drink certain solutions that we today would say contained iron, for example, and this was treating cases of anemia. So when you were very tired and you didn't have enough iron and so on, I mean, people didn't know it was iron at the time, but they realized it helps if you grind up certain salts and you drink these solutions, or you dissolve them in acid, like citric acid or something, and you drink it, you would realize, okay, this was making you feel better, was helping you in your disease state, right? So you can see medicinal inorganic chemistry easily goes back at least 3,000 years. And then this is more or less when science began, in the 1900s, when people were beginning to do careful experiments and they were having a hypothesis, doing an experiment, and then proving or disproving the hypothesis. So this is what we call modern science. Yeah? So when modern science started, you can see suddenly the development also of inorganic drugs started. And bismuth compounds were among the first to be used. Gold compounds were among the first to be used, but now in a rational fashion. They were tried, they were optimized, and then given. Not just like gold amulet, but really like a compound that is given as a drug. And then more than 100 years ago, I'll talk about this compound here in some detail, the first structure-activity relationship ever was established for a drug, and that happened to be an arsenic-containing compound. So this is basically medicinal inorganic chemistry, in a way, started more than 100 years ago. And there are some firsts here, for example, metal-based chemotherapy. Cisplatin, the famous compound, is already, as you can see, about 50 years old. Okay? So we'll go through this, and this slide here will come up again and again, and we'll take it up and take it from there in different compound classes. Just to give you a flavor of what you are expecting and what we are not going to do in this course, in general, we can talk about differences, compounds either being used for diagnosis or for therapy. Those are the two main fields. And in this particular lecture, I will not talk much about diagnosis. I think actually this would be a whole lecture on itself. I've just identified here radio imaging, where you can use a whole zoo of isotopes. Copper is very popular, different isotopes. You can talk about technetium, you can talk about gallium, indium, yttrium, many, many others. All the lanthanides, for example, can be used in MRI or in optical properties. I will not talk about this here. That would be a totally different set of lectures. I will concentrate on therapeutic applications of compounds. I already mentioned arsenic compound. I mentioned bismuth compounds, platinum compounds. We'll spend a whole day. Tomorrow actually will be all about platinum. So if you want to become rich, you have to come tomorrow. Gold compounds will also be touched. That'll be like the day after tomorrow. So it'll be on Wednesday. We'll talk about gold compounds and others like ruthenium, iridium, palladium. So also a lot of noble metals on Wednesday. And then we'll talk on Thursday. We'll touch some of the things that are up here, but not. I'll look at those less from, not compound by compound, but more like from the mode of action kind of point of view. And then you can make up your own mind, and you can actually, if you want to explore new avenues, you can go from there and think about new metal compounds. Okay. Fair enough. So I think actually we can more or less stop here. I just want to highlight, I think I mentioned this already to you, inorganic compounds really make up only a handful of all the compounds on the market. Remember my first two slides when we had this periodic table with just a couple of elements? This is about, in Germany, about 2,000 approved drugs. So if you go into a pharmacy, you have a choice between about 2,000 or so drugs. And only a handful of those, maybe 24 or so, are metal-based compounds. And the remaining, 1,985 or so, are purely organic drugs. So we are talking about a relatively small fraction, but if you see the periodic table, and if you, throughout the week, you'll see the reactivity and the possibilities, then I think growth is probably more in this handful than in those 2,000. You see a couple of examples here. Platinum I already mentioned. Gold I mentioned. This is an iron compound. Antimony, we'll be talking about. Lithium I already mentioned. Arsenic is missing here. Bismuth is missing here. So you could add a few more, but it's really not more than one or two handful of compounds. Oops, sorry. I'll finish by showing you, I hope that works. This is the kind of literature slide that I will have at the end of each lecture. You'll see here sort of some typical textbook and then some more reviews here. Admittedly, for this overview kind of thing, it's really difficult to pick reviews, so I just picked some from my own group that go on, organometallic compounds as anti-cancerous, antibacterial compounds. These textbooks are some that I really liked. I can really recommend. The one here on top by Jim Dobrowiak, Metals in Medicine, is already available in the second edition, 2017, so it's very, very recent. It's actually a very, very good book. You will see that a number of the figures from my slides are actually from this book. I haven't put the acknowledgement in, for each single slide, but I'm putting his book in my literature list all the time, and, as I said, many of the figures will be from his book. Tim Storr has edited. He's actually the editor for this book. He has edited another book, which is relatively recent. And Enzo Alessio, I already pointed out, he also edited a textbook, Bioinorganic Medicinal Chemistry, that is already a couple of years old, but it has some very, very nice chapters as well. And in some cases, I'm actually pointing out the particular chapter in Enzo's book on particular topics like platinum, like ruthenium, and so on. I point those out in particular. And then there is a couple of older ones, a couple of textbook chapters, actually, and you'll see in all the other slides, at the end of each lecture, there's always going to be one literature slide like this where you see some leading references at least. I admit some of it is very, very personal. I mean, it's people I know and reviews that I used. There may be many others in the field if you just search for a keyword and so on. Okay? Okay, I think now it's time to have maybe 10 minutes tea break, and then we come back and I'll go about some more aspects in drug development. Thank you.
drugs, conceptual models in bioinorganic chemistry. And in fact, the last timeline that he had shown about medicinal inorganic chemistry was from last time. So in case anyone is interested, during this week you can have a look at this book, take a photograph, and buy it.
Okay, so welcome everybody back for the second lecture of today. I had some very good discussions and some comments were made. By some colleagues, and I would like to correct one or two things or point out one or two things. When I talked about the number of drugs that are on the market, there was this number, 2,000 compounds in a pharmacy. Of course, I mean, that means just compounds by itself. You can imagine, for example, for something like aspirin, there are many, many generic, makes of aspirin, and it's always the same. It's just one compound, obviously, but it's, available in all forms. I mean, Bayer was the original one, and there is probably 24 or so different aspirin formulations in a pharmacy, and I guess the same is in India. I know actually the Indian government is making a huge effort in terms of drug production and production of generic drugs. I think actually some of the companies are in this area here near Pune, where they actually try to make some of the very expensive drugs that are out there on the market in a generic form for the Indian market to make it available and cheaper, for you guys, so you don't have to buy the expensive stuff from Europe or from the US. Okay, so we were talking about compounds, not about all the different generic makes that there are. I guess then there would be way over, 10,000 maybe or so. So that's one thing to think about. The other thing that one of you pointed out here, talking about lithium, and I'm not going to touch lithium any further, was just one example, but lithium really works as a lithium ion as it comes. And if you chelate it or if you put it into some larger complex, then it actually loses its efficiency. So it really has to be the lithium ion. And the lithium ion seems to interact with some membranes, and that's why you need this very small, singly positively charged ion that interacts with the membrane and changes the membrane structure, and then that helps cure depression. Now, the one warning here is this really only works if you are ill, if you are depressive, and even then it doesn't work for everybody. So please do not take a spoonful of lithium every morning. Yeah? Lithium does not make you high, okay? It's not a psychedelic drug or something. You have to smoke hash or something, but don't eat lithium, okay? Okay, just a warning, not that people say, I recommended eating lithium and you feel good. That does not work. Okay, all right. Talking about things that you should not do. Don't eat, whatever, lithium or platinum or whatever. We'll talk a little bit about toxicology, and I'll talk about things that happens when you don't have enough of something. That should, of course, be deficiency. I'm sorry, there's a mistake here, typo here. But we'll talk a little bit about toxicology. So this is the side of things when things actually get... So toxicology is when things actually get bad, when you're getting too much of it or when something is actually harmful to it. And you will see that there is a fine line sometimes between something being toxic and something being beneficial. And we'll talk about that, and we'll talk, of course, because we are scientists, we'll talk about it in a qualitative and in a quantitative way. So we'll put numbers to it, and we can actually quantify these aspects. So this is a table of the elements. It's now completely filled up, and it's color-coded. And the green elements in this table here are the ones that you absolutely need as a human. So you'll see a lot of the transition metals from the first transition metal row, starting by vanadium, going all the way to zinc, are actually essential elements for humans. So you definitely need those. You will also notice that molybdenum is the only transition metal from the second row here, which you need. There's a question mark behind chromium, because if you look at my body, at your body, you will find chromium in my body, in your body. So you can think, oh, okay, chromium is important. But nobody yet has demonstrated a function where chromium is actually needed. nobody knows whether there is an enzyme that naturally has chromium in its active site. There may be one, it's just not yet discovered, yeah? But maybe it's only that you and I and everybody has chromium in our bodies because it is so abundant. Yeah? There is chromium in food, there is chromium in your drinking water to some very, very small quantities, there is chromium in God knows what, I don't know. I mean, maybe something you take in, there may be chromium. Another case is titanium, for example. Yeah? Titanium is everywhere around you. It's in the color in your wall paint. It's in your toothpaste. It's an ingredient in many, many things that you eat. Yeah? Actually, I even heard there is apple juice. If it's the turbid apple juice, which in Germany is very popular because you think, it's not filtered, so it's pure. It's like sort of more natural. The opposite is true. They take the filtered apple juice and they add titanium dioxide to it to make it turbid and to sell it to a higher price. So how stupid is that, right? But you see, we are eating it, we are drinking it. I mean, you don't eat your toothpaste, but my children, when they were young, they used to eat their toothpaste. So you are eating and drinking titanium in the form of titanium dioxide all the time, which means there is titanium in your body and my body, but it's not essential. It's there, but it doesn't have a role. So the green elements here are those which are in your body and my body, and for those has been demonstrated a function. So if they're missing, you really go ill. Okay? So that's where the color coding from. Chromium is in your body and in my body and in everybody's body, well, more or less, I guess. But it's not clear whether there is a function for chromium. There may be. I mean, if you're a smart scientist, maybe you will discover one. You will be the one to discover this is really the essential chromium enzyme. As far as I know, nobody has demonstrated that so far. Here is the elemental composition of the human body. And this is interesting from an inorganic point of view. The most abundant metal in your body and my body is by far calcium. Yeah? There's about 1 kilogram of calcium in each of you. There's a lot of calcium in milk. And the analogy I used to make in my home university, we have these liter bottles of milk, So there's like a liter, like a kilogram of milk in it. And I keep saying, this 1 kilogram is actually calcium in your body. Milk contains a lot of calcium, if you know that. So this is something that children or also the students can always remember. So 1 kilogram of calcium is in your body, and all the other ones, like potassium and sodium, a lot less, 100 milligrams. 100 grams, not milligrams, 100 grams. But calcium is like 10 times more. So that's really, really massive. Who knows what is the most abundant transition metal in your body and in mine, if we're healthy? Iron. Yeah, very good. Excellent. How much iron is in your body and mine? 5 grams. Okay? 1 kilogram versus 5 grams. So that's not a lot. Here's the numbers. Iron is up here. Okay, 4.5. No, never mind. I mean, there's plus minus something. There's an error on this, right? So this is already milligram scale. Iron is the most abundant transition metal in your body and in my body. Very important. Everybody knows this. Zinc is the second most abundant one, about half as much, is about half the amount of zinc than iron in your body. And then all the other ones, copper, manganese, chromium. You see, this is 5 milligrams of chromium is in your body. This is suggested, it's supposed to be connected to glucose metabolism, but nobody has yet demonstrated the one crucial enzyme which contains this. Therefore, there's a question mark here. Chromium, cobalt, molybdenum, all the other ones here, milligram amounts, almost nothing. Milligram. My weight is 70 kilos. When I'm too long in India, it's more like 73 because I eat too much and the food is so nice. But normally, let's say 70 kilo, milligram. Cobalt, 3 milligram of cobalt in 70 kilo. You try and find this. It's really difficult to isolate. Even, your trace analytics have to be really good. And look at this. It has already been identified in the 1930s that cobalt is in your body, in my body, and it has a vital function. So these were really, really smart scientists, to find 3 milligram in 70 kilo. That's pretty good. And I'm seriously, I'm admiring this. Yeah, this is really, really good in the 1930s. I mean, they didn't have an ICP-MS or anything like this. They were just doing elemental analysis. Okay, in comparison, iron is relatively easier. 5 gram you should be able to find. Yeah? If you're a good analytical scientist, that should be possible. Okay, fair enough. So let's go on about some of these things. I would like to talk about, when things are good, when things are bad about you. We talk about metal deficiency. So if you don't have enough of it, iron, of course, I told you, has been known that if you are lacking iron, you feel ill. And it has already been known, before the 1800s, that it was good if you, try and grind up certain salts that were rich in iron, of course, some rust, basically, iron-containing minerals, and you drink them, you systematically drink them, then it helps you feel better. And the first documented case of a metal deficiency in terms of a scientific investigation, in fact, was zinc. And this was in some place in rural Iran. So where it was clear that people had deficiency. They were relatively small. Sexual development would be delayed for quite some time. And this was traced back to low levels of zinc in the blood of these people. And if you then just supplement zinc salt just by making them eat zinc salts like zinc chloride or something, then it would help relieve those symptoms. And nowadays it's not a problem anymore because in this area there's simply supplement in the food, zinc, and then people are perfectly okay. And this was the first scientific study more than 50 years ago when this was established. So what we are talking about here is metal-ion homeostasis. Homeostasis means, it's the definition, that you want to maintain the correct concentration of a certain metal or metal ions in your body. And you can do this either by regulating the intake. So if you don't have enough of it, you take more in. Or you can do this by regulating the excretion. If you don't have enough of it, you limit excretion, and then it accumulates, and you get back to normal levels. So you have either way of playing around with it, uptake or excretion, and you want to get the metal concentration in your body just right. Just briefly talking about zinc, and this is something you certainly don't have to remember, but I just want to give you a flavor of what people are doing. Nowadays, we talk about active uptake of zinc into cells. So there's a zinc transporter. These are zinc transporter enzymes. At least 3 of them are known. There is some intracellular machinery, for example, in big size. So they could tell there's something wrong with the red blood cells, but they didn't know what it is. And it was discovered basically by accident that raw liver extract would help cure this anemia. I mean, it's pretty nasty, right? If you're made to eat or drink raw liver extracts, I don't think you want to do that. But anyway, it was discovered that this was help the children. And so a Nobel Prize at the time was awarded for these very careful, thorough investigations on these treatments. But of course, nobody knew, what was behind it. It would help, but you wouldn't know how. So what came out in the 1940s is that there is a crystalline red compound in this raw liver, and that was isolated, and this was actually the active compound. So you could take this compound and give it to the children, and they would be cured as well. So you don't have to take raw liver, which is a big advancement, I would say. No? The crystal structure of this compound was solved by Dorothy Crawford Hodgkin, a very famous crystallographer, and it turns out this is vitamin B12. That's the compound that was made. And you will see the structure in a second. This was a very, very difficult structure to solve at the time in the 1960s without the help of any computers. You'll see it's a very complicated molecule. And again, a second Nobel Prize was awarded in this field to Dorothy Hodgkin. And then later on, the total synthesis of vitamin B12 was reported. And then there is more crystal structures here of vitamin B12-dependent enzymes, and so on, and so on, and so on. And the whole field still continues, and it's more than 100 years now. This is the woman we are talking about, Dorothy Crawford Hodgkin, a very, very good scientist, a crystallographer, and she used to work in Oxford. And I'm just showing this slide because there is a small, it's called the Crystallography Lab, which is basically really a very small, it's more like a hut, like a sort of small white building. This is where she used to work, and it's still the place where all the diffractometers in Oxford are placed. Yeah, and I have a crystallography department. Basically, you can almost see her office now still. It's still the same furniture and so on. Oxford is one of these universities which, where change comes very, very slowly only. But they are 600 years old, so maybe they don't have to change very much. Okay, this is the structure of vitamin B12. Maybe it's familiar to some of you. Maybe you have heard of it already? Yeah. Okay, so I don't need to go into great details. There's a cobalt in the middle. It has this chlorine ring system. Very strange cyclic structure. The really important thing is these R residues here. It can be cyanocobalamin or methylcobalamin or this adenosylcobalamin or even a hydroxycobalamin. And the 3D structure is this one here. And I think there is a lot of animations that we don't have to go through. I guess the really important thing is these cobalt to methyl bond in methylcobalamin or adenosylcobalamin. That's the really unusual feature. So in a way, cobalamin is actually an organometallic compound. Methylcobalamin or adenosylcobalamin here. This is the crucial thing. And it does some very peculiar chemistry, as you know. Cobalamin can react as a carb anion or as a carbocation or as a radical. And this, of course, has to do with the cobalt chemistry. It can be cobalt-free, it can be cobalt-2, or cobalt-1. And all three oxidation states are available in biological media. And depending whether it's a cobalt-free, then you get a carb anion, or it's a cobalt-2 and a radical, or a cobalt-1 and a carbocation. So depending on the oxidation state of the cobalt, the carbon atom next to it has totally different reactivity. So this is really, really very versatile, and it's used for a number of things like methylation, for example, of amino acids, of DNA, like transmutation reactions or methyl transfer reactions. So this is really, really versatile, has some very interesting chemistry. If you think about cobalt uptake, for example, getting enough cobalt into your body, this is a very overview type pictures. You can have the cobalt B12, vitamin B12, so-called coenzymes. So this could be, for example, the hydroxycobalamin or the cyanocobalamin. And then it's taken up into the stomach. And there is some so-called, this is the extrinsic factor, and some so-called intrinsic factor, which is like a vitamin B12 recognition enzyme. And they together, they take the vitamin B12, which is in your food, into your blood cells. So this is a blood cell, and this is your food. So you got intracellular B12 now. And it's transformed into hydroxyvitamin B12, methyl B12, which is, for example, important for biosynthesis of amino acids. So if you have a new cell that is growing, it needs to synthesize amino acid, it needs vitamin B12. There is also a DNA biosynthesis path here. And then if you talk about deoxyadenosyl B12, this transforms methylmalonyl into succinyl-CoA, and this again is important for fatty acid metabolism. So you have fatty acid metabolism, DNA and amino acid biosynthesis. Everything is coupled to vitamin B12. You can imagine if there is no vitamin B12, things go horribly wrong. Amino acid metabolism stops, lipid metabolism goes wrong. So this is horrible, and this is what's happening, what the disease is. Now, the reason for this pernicious anemia is that there is an autoimmune disease which develops antibodies against this intrinsic factor or the complex of the intrinsic factor of vitamin B12. So you can see this uptake mechanism here. This uptake mechanism goes horribly wrong because an antibody recognizes this and catches this, so the vitamin B12 doesn't get into the blood. So it's an autoimmune disease where you produce this antibody, which you shouldn't produce in the first place. Should never be there. And this is a fatal disease. It's diagnosed by this typical appearance of red blood cells. Now, you really have to look at the numbers. We are looking at very small numbers. Your body and my body, as you saw, contains a few milligrams of cobalt. As we said, 3 milligrams, which is a really small amount. If you eat normal, you eat like fruit and maybe vegetables and maybe some meat or maybe not, doesn't matter really, you have more than enough vitamin B12 in your food because basically it's produced by bacteria. Your salad doesn't produce any vitamin B12, but the bacteria which are sitting on the salad, they do. So you're getting the vitamin B12 from all the bacteria that you're taking up inevitably. It's quite fine, it's no problem with it. You're getting more than enough vitamin B12. You resorb only a few micrograms. You see, this is tiny, tiny amounts. And the uptake is slow. But these few micrograms, it's more than enough to supply all the cobalt that you need. Cobalt is excreted in very small amounts, just like the normal way. You're sweating, you're going to the toilet. So this way, you lose a few micrograms of cobalt every day, and you're taking up a few micrograms as well. And this maintains homeostasis in your body, and you need this in the form of vitamin B12. So when you have this disease here, you don't get those 5 micrograms anymore. But what you can do is you can basically just eat more vitamin B12. You take vitamin B12 as pills, or it can be, injected. You can take it parenteral vitamin B12. You're getting many, many more. You're getting maybe, a couple of milligrams or so per month, as opposed to a few micrograms. And then this just increases the number of vitamin B12 that is available. It swamps the system. And in this way, you can trick this autoimmune disease, you can trick these antibodies by just supplying so much of the vitamin B12 that this system simply is, you can override the system, and you get enough vitamin B12 in your body, and you're fine. It will be cured. And this is especially good, as we saw, for children, because previously we used to die around the age of 10. When you are born as a child, you actually get those 3 milligrams of cobalt from your mother. Yeah? So the children are well equipped with enough cobalt. And this lasts for a few days because you're only excreting maybe a microgram or something per day. Very, very little. Yeah? And in this sense, the milligrams of cobalt that the children had lasts a few years. And then if they have this disease, it doesn't get supplied, so they really die because they have finally lost all the cobalt they had from their mother. Okay? But simply by eating vitamin B12 pills, you can go to a pharmacy, you can buy it, you can eat it, and you're perfectly okay. So this brings us to a scientific question. How much do we need to take in? And if we eat more, if you would eat vitamin B12 or I, would it be harmful? Would we suffer? And the answer is no, because this uptake here is regulated. Even if we take much more vitamin B12, we would only get these couple of milligrams, so it wouldn't be harmful. But in a scientific way, this is a famous so-called Bertrand Diagram from a French scientist, Bertrand. And this basically shows you there is an area where you raise the concentration of either a drug or a metal or something else. If you have not enough of it, you suffer from deficiency and you're suffering. Yeah? So when the curve goes down, it means you're suffering. When the curve is up here, it means you're well. And then there is a range which is good for you. This is what you need. But if you get too much of it, it's toxic. And we'll talk about some toxic metal compounds here on Friday, I think. So there's going to be more details on this part here. We'll talk a little bit about this part here and about this part here. The safe dose, when are things actually in the right range? There is a famous saying by sort of a medieval healer, let's say, Paracelsus, who was very, very famous in medicinal chemistry because he was like a guru or so of medicinal chemistry in those ages. And he said some very, very true things. He wasn't a scientist because science wasn't really born at that time, but he said some very true things. For example, he says that the dose is what makes things a poison. And you all know this, if you take a little sip of whiskey, that's not harmful, but if you drink a whole bottle of whiskey, it's toxic. So, you can clearly align with Paracelsus. I don't think he was talking about whiskey, though, but anyway. All right, okay. Historically, of course, Paracelsus or people in this field, they devised, actually it was Paul Ehrlich who devised this. Today we use something that we call a therapeutic index. And you have to determine how much is lethal. So when does, for example, an animal die? And what dose is required for therapeutic effects. So this is the efficient dose when 50% of your patients are healed, or let's say 50% of your animals survive. And then as opposed to the so-called LD50, which is a lethal dose when 50% of your test animals die. And you can imagine if the LD50 and the ED50 are very similar, yeah? So you need a relatively high amount to cure the disease. On the other hand, this relatively high amount has a high likelihood of killing your animals, your test animals. Then this is a very dangerous compound to use. So what you want is something where you have a low dose for activity and a very high dose is needed to actually kill the animal. So then you have what we say is a very safe drug. Yeah? Even if by accident you take twice the amount because you don't remember, did I take my pill already? Yes or no? I'd rather take a second one. So that should not be harmful. Yeah? So this is what we call the so-called therapeutic window or the therapeutic index. Yeah? It's a measure for drug safety. If we look at typical metal compounds or metal ions, we can classify them and say, what about, these compounds that we saw, what about their therapeutic window? For example, for some toxic metal ions, like mercury, for example, there is no therapeutic windows because there is no beneficial effect. Ideally, you want 0 mercury in your body and in my body, right? Any mercury that you're getting is too much, period, end of story. No therapeutic window. Yeah? On the other hand, you may be amazed to hear that things like molybdenum and chromium, actually, well, chromium, little question mark here, okay, whether it's beneficial or not, but molybdenum, you can take relatively large doses of molybdenum. It doesn't harm you. You need a little bit, fine, but even if you take, 10 times of this or 100 times, it's still okay. You won't suffer. Interestingly, for example, iron, this is not the case. We talked about iron. We know that there is 5 grams of iron in your body and in mine. If you have 10 grams of iron, twice, so the therapeutic index would be 2. 10 divided by 2. Well, you don't die from 10 grams. But anyway, if you have 10 grams of iron in your body, you will already suffer. If you have 15 or 20 grams of iron, you will have these sort of dark marks under your skin. Basically, it's rust. You have iron oxide under your skin because it's deposited. And it's not just deposited under your skin. You could say, well, who cares? Most of you have brown color anyway, so what about a little bit of rust? But it will deposit also in your liver, for example. And then, of course, your liver is damaged, right? So iron, surprisingly, you need 5 grams, but 10 grams, therapeutic factor of 2, is already too much. So iron has a relatively small therapeutic window, surprisingly. And then, for example, there are things which are like bismuth compounds, for example. I mean, we'll talk about bismuth a little bit. It turns out bismuth has a very high therapeutic index because you can take, 10 times or 100 times the dose of what you were prescribed. It doesn't really harm you. It just leaves your body right away. So it doesn't harm you. Again, for example, cobalt salts. Thinking about vitamin B12. We talked about vitamin B12. Cobalt salts by themselves have no benefit. So cobalt has no therapeutic window. There is no dose-response curve. Because you do not know cobalt. You can eat cobalt if you have pernicious anemia. You eat cobalt, it has no effect because you need vitamin B12 and not just cobalt salts. Because the vitamin B12, which you eat and which I eat, comes from bacteria. We, ourselves, we cannot make vitamin B12. No way. We have to take it with our food from the bacteria. So we need the bacteria who make it. Which means eating cobalt or not eating cobalt has no effect. If you have pernicious anemia, that's not enough. You really need this one single compound. Whereas vitamin B12 has a very high therapeutic index. I told you, if I take vitamin B12 pills, it has no effect on me. It's not harmful, it's not good, it just doesn't matter. So don't spend money on vitamin B12 pills. It has no effect, it doesn't matter. Okay. If we talk about toxicity, there are just a few, we just need to be clear about a few definitions, and this will come up throughout the lecture. There will be sort of certain points that we discuss. There are a few definitions here that we have to be aware about. Sometimes when we talk about, for example, an anti-cancer drug, we actually want a drug that is toxic because the anti-cancer drug should kill the cancer cells. So yes, we want toxic compounds. Brackets, in certain cases. So toxicity itself is not bad. And it's not just about cancer. It's not just that, we need, for example, a compound like cisplatin, which is toxic to cells. But we also sometimes need something that is species-specific, has species-specific toxicity. For example, if we think about antibiotics, of course the antibiotic must be toxic to bacteria. And it should not be toxic to you and me. That's the one requirement in an antibiotic. Be safe for use in humans, no effect on you and me, but yes, of course, be toxic to the bacteria. Kill the bacteria, all of them, immediately. So yes, we want toxic compounds, but we need to be careful about what we define as toxicity. And as we said, it can be, toxicity against microorganisms, antimicrobials, for example, things like this. And in fact, some of the early metal-based antimicrobials were in fact toxic against humans as well as against bacteria. So they were not good antibiotics in that sense, and that's why they phased out. Something like penicillin, for example, well-known antibiotic, is actually a very, very safe drug, because even if you take large amount of penicillin, usually it doesn't affect you. Yeah? So penicillin in that sense is a very good drug because it's safe for us to use and it's toxic against bacteria. This is relatively easy to achieve if you think of it, because the bacterium has a totally different metabolism. It has a different cell wall to start with. The bacterium has no liver. Yeah, it's a totally different organism. So there are many, many opportunities to be very specific against a bacterium without harming you or me. But if you talk about cancer, for example, this is a cell that is identical to the cells in my body as well. So there are only some tiny genetic differences. Maybe it's even, metagenomics. Some methylations that are different, some metabolism that went wrong at the wrong time. Yeah, but it's still a cell identical to the other cells in my body if I had cancer, which I hope I don't have. So here we talk about toxicity against a certain type of cells. If we have an anti-cancer drug, we only want to kill the cancer cell, but not the normal cell, which is directly next to it. You can imagine how difficult this is. You have to identify something that's different in the cancer cell that we can target, that is specific for the cancer cell, but of course not for the normal cell. This is totally difficult. Yeah, very, very difficult task to achieve. We will talk towards the end of the lecture, we will talk about some strategies, so-called targeting of specific cell types, yeah, to really treat cancer and not the normal cells. It's not easy to do. It's not an easy thing to do. We'll talk about some aspects how we can achieve this. This is what people call a dose response curve, and I'm sorry this is in German. I didn't find in English a good English version of it, but you can still read. This is toxic. You can still read that, right? And that's lethal. So I guess even if you learn 2 words of German now, okay? Right? So you see curve here. This is the percentage of people, yeah. And of course, if you have a very low concentration, nothing happens, and then you raise the concentration and you get some toxic effect here. And then at an even higher concentration, you get a lethal effect. And this is here when people die. The LD50 we mentioned before, this is the lethal dose, and this is the toxic dose, as you can imagine. So again, you have something like a window here. It's not therapeutic, but it's a window. And you have a first toxic effect, and then when people die. And so these are some basics here again. And of course, for any drug, you want this lethal dose, you want to be as high as possible. Yeah, the higher the lethal dose, the more effective or the safer your drug is. There are some important parameters that we just need to take into consideration. We've been talking a lot today about drugs and usage of drugs, optimizations. We talked about pharmacokinetics and ADME. You remember absorption, distribution, metabolism, and so on, excretion, right? These things are important in relation to it. For example, the question is, how do we apply the drug? Yeah. Is it by injection, for example, or is it through the skin, which we would say dermal, or is it injection under the skin, not into the veins, for example, intravenously, or just under the skin? And all these things, of course, have to do with uptake. And if you inject into the veins, into your blood directly, of course, the drug is there immediately, but it may be too much, yeah, at that moment. Or maybe there is a metabolism in the liver which you don't want. So you just inject it under the skin, and then the drug is very slowly absorbed into the blood, yeah, and it's distributed much more evenly. So all these things are important when we talk about uptake, when we talk about metabolism, when we talk about bioavailability. It may be that you might have something that is actually quite potent and quite good, but when you're applying it the wrong way, it doesn't work. And the typical example is, what you would like to have is oral uptake. Yeah? Basically, if you have a drug, you just want to take a pill. For example, if you need an injection, you need to go to a doctor. Very often, you actually need to go into a hospital. Who wants to go to a hospital? Nobody, right? So you really want to have something that is orally available. Now, unfortunately, all the very good anti-cancer drugs, which we will talk about tomorrow, cisplatin drugs, none of them is available orally. You have to go into a hospital. If you need to be treated with a platinum drug, it absolutely requires at the moment that you spend time in a hospital. You can say, okay, like in a city like Bochum, there is many hospitals, so it's maybe not such an effort. But if you think about rural India, yeah, you think about transportation in your country, where's the next hospital where you can get such a treatment? So clearly there's really big issues here, and ideally you want oral administration, but for the drugs that we will talk about tomorrow all day, none of them is orally available. But of course there is a lot of need for research here to make such drugs orally available, because that would give, potentially at least, it would give treatment to many more people and much more convenient. Okay. Talking about, toxicity, I talked about different species already. We don't need to go into details here anymore. The other thing I just briefly wanted to point out is, of course, that if you take a drug, it's never evenly there. You take the drug, it's taken up into your blood. For example, if we talk about subcutaneously, then it actually takes a very long time until the concentration of your drugs reaches something where you see at least some minimal effect here. This is minimal, okay? You're learning a third word of German. This is maximal, okay? So 4. You learned 4 words of German in this lecture. So here you can see, it takes some time, latency period, until you're actually getting an effect. You know this very well. If you take, if you have a headache, you take aspirin. How long does it take until you have an effect? 15 minutes or so? 20 minutes? Yeah. Ideally, of course, if you have a headache, you take a pill, you want, within 2 minutes or 1 minute, you want to have an effect. That's usually not the case. For aspirin or ibuprofen or so, for me, it takes 15 minutes or something at least. Yeah? And then, of course, if you have a very bad headache, while you're okay for a while, and then the concentration of the drug in the blood goes down again, and you lose the effect again. So if you take 1 aspirin, let's say, after 2, 3, 4 hours, the headache comes back, because the aspirin is degraded in the blood, it's taken up, it's degraded in liver, you don't have an effect anymore. So you need to take another aspirin. Yeah? So this is something to keep in mind. It may be, for example, that this takes very, very long until you actually see an effect. We'll talk about toxicity, for example, about mercury compounds. We will see that this period can be a few months until you see an effect. When you have mercury poison, acute mercury poison, this is super dangerous because you don't feel anything right away, but you suffer in 3 months' time or 4 months' time. And then it's already too late. I'll give you an example on the very last day. Not a very pleasant one, but just to make you aware of this. This can be very, very long. Aspirin, 15 minutes. Not so much of a problem, okay? But of course, things are degraded, so, it goes down again. So at this moment, at least, but probably here, you have to take another dose of the drug, so you get the next curve here and then the next one, so on and so on. Ideally, of course, this is now 25 hours, so this is like 1 day, if you like. Yeah, so this would be a drug which you take once a day. But sometimes you have to take 3 doses a day, or sometimes you have to take a dose every 5 days or so, and this depends, of course, on the metabolism of the drug. We talk about acute toxicity and chronic toxicity, which takes, following a long intake, or some toxicity that comes up very quickly within a day or so, basically. Okay, so we have some fundamentals of metal deficiency. We had some dose response curves today. We had, this Bertrand diagram where you talk about deficiency, beneficial effect, toxicity, and we'll be moving in that window. And we also learned that toxicity by itself is not a bad thing, so we have to direct it. We have to be toxic to the right thing, species or cells, and we'll talk more about that tomorrow. We'll treat anti-cancer compounds tomorrow. We'll start by cisplatin, the very famous one, and then we'll talk about some modern developments. So I'll take you right from the discovery up to what is research state-of-the-art now. Well, now meaning, in recent years, you'll get some information. I'll finish with this lecture. As I said, there are some references here. This is all very, very general. Or what you find here is a lot of knowledge that is basically general knowledge in textbooks about toxicology, medicinal chemistry, and so on. And we'll be more specific about one particular class of compounds tomorrow. Have a very nice evening. I hope to see all of you again tomorrow. Thank you.
Any questions? Anyone have?
Yeah, sure. I'm sorry. Yes. No, go ahead. Yeah, we actually should have a session, at least the question section, maybe after each session, right? Yeah. I mean, I didn't see any hands going up during the lecture. I was looking, but there is a question at the back. Go on. Shout, shout, I can't hear you.
Hello, hello.
Okay. How does the body remove excess metal? That very much depends. This depends on the metal. For some metals, actually, there is an active mechanism when you have, like you have transporters which take things into the cell. There is a similar transporter system which is for excretion of the cell, of the compound from the cell. For example, this exists for copper. If you have too much copper,